The world around us is rapidly changing and the sea level is rising. Due to sea level rise, we need to think about spatial planning adapted to climate change consequences. One of the solutions which has gotten an increase of attention is floating structures such as offshore solar panels and floating neighborhoods. Floating foundations allow water to provide viable new building space for infrastructure and a whole new world of possibilities. Our researchers aim to understand what benthic microflora and fauna will grow on these structures depending on the depth and submergence time, and in our case we are using wood as a substrate. Our research question is, how do the types of communities differ on wood in the Wadden Sea depending on the depth and submergence time? Then we have 3 sub-research questions. Our first one is, how does the biomass differ on wood in the Wadden Sea depending on the depth and submergence time? Number 2, how does the species richness differ on wood in the Wadden Sea depending on the depth and submergence time? And number 3, which species characterize the benthic microflora and fauna on wood in the Wadden Sea depending on the depth and submergence time? Our hypothesis is that we are expecting to find many oysters and types of seaweed when snorkeling in the sublittoral zone, and in the middle tide zone we are expecting to find mostly mussels and barnacles, while in the high tide zone we are expected to find many small flora and snails.
Our sampling location on Texel was Watersportvereniging Oude Schild, next to the harbor. We took samples on 3 different heights with 3 different submergence times. For each height we took 3 replicates. The first set of samples were taken from the side of a floating jetty. As you can see in the drawing, these samples were submerged 100% of the time. The second day we went snorkeling during low tide. We took samples at a height of 44 centimeters below NAP. This is the top of the sampling frame. This set of samples was submerged around 70% of the time. The third day we took samples from shore at a height of 12 centimeters above NAP. These samples were submerged about 40% of the time. Now for the actual process. Each time we used a frame of 32 by 16 centimeters to mark the area that we wanted to sample. We marked the area by scraping the borders of the area clean. Then we used a putty knife to scrape the benthos of the marked area. We collected the benthos in a net while snorkeling and transferred them to plastic boxes for transport to the lab tent. We also added some seawater to these boxes to keep the samples fresh. For the samples we took above water, we collected them into plastic boxes immediately. Of course, we used some materials and took some safety measures. We used wetsuits, diving shoes, working gloves, goggles and a snorkel, and of course Leia was there for our safety. We used a raster, a putty knife, a net with small mesh eyes and tie wraps to take our samples.
Once we got our samples in the lab, the endless job of identifying our samples started. Firstly, we sieved our samples in a sieve of 1 mm and put our samples in sorting trays. We used tweezers to take our samples apart and identified and counted them with the help of books and identification keys. After identifying our samples, we put them in plastic bags and put them in the freezer. We made sure to be careful with labeling our samples. Back in Wageningen, more identifying and counting of sample awaited us. Here we had access to better materials. We sorted our samples into aluminium trays to put them in the oven to dry at 105 degrees Celsius. We had about 60 trays in total. Once 24 hours had passed, we took all of our samples out of the oven and weighed them again. Afterwards, we put the mussels and the oysters in the ashing furnace on 550 degrees. This oven is used to burn all organic matter, to get a clear picture of the actual biomass of the mussels and oysters.
As explained before, we took our samples at 3 different depths. This gives us 3 conditions. The first condition is a condition where the benthos are generally always submerged. This one will be known as the always submerged condition. The second one will be the 70% submerged condition, and the third one will be the 40% submerged condition. The always submerged condition was home to a variety of species, including many barnacles, mussels, and a variety of algae. The 70% and 40% conditions were home to mostly green algae and barnacles. We measured the total biomass of all macrobenthic organisms found on the different levels of depth and used an ANOVA to test the statistical significance. We used a significance level of 0.05 and our data met the assumption of normality and the assumption of equivariance measured with the Levene test. The results showed that the total biomass of our 3 groups differ from each other significantly with a p-value of 0.000457. A post-hoc analysis using the Tukey test showed us that the different groups differ from each other significantly as well. We encountered similar results in our evaluation of species richness. First, we ran an ANOVA to calculate whether there was a difference between the 3 groups in terms of species richness. We used a significance level of 0.05 and our data met all necessary assumptions. We identified the algae on division level unless they were easily identifiable on a higher taxonomic level. The analysis gave us a p-value of 0.00268, which means there's a significant difference in the species richness between the groups. We used a post-hoc analysis to look at the differences between the groups, and the results showed a significant difference between the always submerged condition and the two other groups, but not between the 70% and 40% submerged conditions themselves. This shows that the always submerged condition differs significantly from the two other groups in terms of species richness.
Okay, let's rewind for a bit. The research question that we've been trying to answer in this video is, how do benthic communities change with different depths and submergence times on wooden surfaces in the Wadden Sea? The objective of this research is to be able to predict what kind of benthic communities will be found on floating structures, varying from jetties to floating cities. So, what can we conclude about this? 1. Benthic life on wooden substrate varies greatly depending on depth and submergence time. 2. There appears to be a positive relation between the biomass and submergence time in a near littoral zone, i.e. the more area submerged, the more biomass. And 3. While adapting our infrastructure to climate change, it is important to keep in mind that communities may differ depending on depth and submergence times. The Wur does not have the permits we need to take samples at the location of our original research, so we had to change our plan entirely. We were very dependent on the tide and the wind. Some parts of our samples may have gotten lost due to the wind, the current, or the transport. Identifying the smaller organisms and algae proved to be very difficult and time-consuming, so we chose to group the small organisms together and subdivide the algae into 3 groups, red, brown, and green. For both the algae and the smaller organisms, this means that we didn't identify them to species level, but we did consider them for the total biomass. Our samples for the category 100% submerged were taken from the side of a floating jetty. This means that the samples were always close to the surface. And last but not least, everything took longer than we originally imagined. This once again called for adaptations to our plan, which I think we handled pretty well.